Hello, my name is Manuela Izquierdo. I'm from third semester of medicine, and today I'm going to talk about this article called Wuzhi capsule attenuates liver steatosis and inflammation during non-alcoholic fatty liver disease development. So, in order to understand the subject, we have to do a little bit of an introduction about what the Wuzhi capsule is. It is an extract from a plant called Schizandra spinathera. This has been used as a medical herb due to its numerous pharmacological uses, such as improving organ function, increasing physical working capacity, normalizing blood and sugar pressure, and also boosting recovery after surgery. On the other hand, we have the non-alcoholic fatty liver disease, which is going to be addressed from now on as NAFLD. This is an abnormal accumulation of lipids due to causes other than alcohol use. There are no specific causes, but of course there are some risk factors such as high cholesterol, hypertension, obesity, and all of those that conclude into metabolic syndrome. And the result of this disease is, of course, the accumulation of lipids, and that leads to inflammation and fibrosis of the liver. So, this study proposes the interaction between the Wuzhi capsule and how it can target the liver by reducing inflammation and steatosis, by decreasing ALT levels, which is an hepatic marker of damage, decreasing interleukin and tumoral necrosis factor levels, which are pro-inflammatory cytokines, and also by increasing PPR levels, which are needed to reduce triglycerides levels. Objective: Wuzhi capsule contains an ethanol extract of Schizandra spinathera. The efficacy of Wuzhi in treating non-alcoholic fatty liver disease has not yet been elucidated. The present study assessed the effects of Wuzhi on NAFLD. For materials and methods, they used a group of mice. Mice were grouped into 10 groups, and each group had 8 mice. They were fed with a basic diet called the MCT diet. The MCT diet is a diet deficient in methionine and choline. These are amino acids needed for the assembly of very low density lipids. Therefore, there's going to be an accumulation of cholesterol and triglycerides. So in general, what this diet does is that it induces the disease to the group of mice. So from group 1 to 6, they were fed with the MCT diet plus the Wuzhi capsule in different doses. For group 7, there was the control diet, which is a diet that has these amino acids, and with the vehicle, which is saline solution. For group number 8 was the control with the Wuzhi capsule. Group number 9 was the MCT diet and the vehicle, and finally group number 10 for verification, again with the MCT diet and the Wuzhi capsule. Mice from group 7 to 10 were sacrificed after feeding them for 5 weeks. Their sera were isolated and the livers were excised rapidly and stored for later analysis. ELISA is an assay technique used to detect and quantify an antibody or antigen. It relies on antibodies to detect a target antigen using highly specific antibody-antigen interactions. And in this study, it was used to detect concentrations of interleukin-1 beta, interleukin-6, tumoral necrosis factor, and interleukin-10. PCR was also used. Polymerase chain reaction allows a specifically targeted DNA sequence to be identified and quantified. The amplification of the DNA is detected by a fluorescent reporter, and in this study it was used to determine the total RNA expression levels of interleukin-1, interleukin-6, tumoral necrosis factor, and interleukin-10 in hepatic tissues. Western blot was also used to evaluate the reaction between an antigen and an antibody. This reaction is done in electrophoresis, and the antibodies are marked to evaluate if it's positive or negative, and it was used in order to detect total protein. Polyclonal rapid anti-mouse PPR alpha, PPR gamma, P65, lamin B, GAPDH, MCAD, and LCAD were used as primary antibodies. Finally, tunnel was also used to detect DNA fragmentations in apoptotic cells by labeling a specific part in the double-strand DNA breaks that are generated during apoptosis. And this was done, of course, to identify the quantity of tunnel and DAPI-positive nuclei. Results. So in here we can see an histological sample. So there is something that has to be clarified that's going to repeat in all the results from now on, and is that these two groups are going to be very similar and are going to be the healthier ones because none of them have the MCT diet. So we're always going to center our attention in these two groups that were fed with the MCT diet, but one has the vehicle and the other one has the Wuzhi capsule. In here we can clearly see that there are more white spaces which means that here the tissue has more fibrosis than in here. So we can conclude that with the Wuzhi capsule, we're going to have a healthier tissue than without it. And in here, they're showing us the same thing, and it's just a different coloration that targets fresh tissue. In this result, they are showing us a double staining of liver sections with monoclonal antibodies. In the first quadrant, it's against DAPI and CD68. In the second quadrant, it's against DAPI and myeloperoxidase. And finally, in the third quadrant, it's against DAPI and tunnel. For these two, it's going to be a little bit hard to determine because they're similar. But for example, for CD68, which was used to determine macrophage activity, we can see that the group that didn't have the Wuzhi capsule is going to have more macrophage activity. Therefore, it makes sense that the group that has the Wuzhi capsule is going to have less of this activity. But where we can really see a difference is in the tunnel result. Tunnel was used to detect apoptotic cell activity. Therefore, we will think that with the Wuzhi capsule, we would have less apoptotic cell activity, and with the group that didn't have the Wuzhi capsule, we will have more apoptotic cell activity, and that's what we can prove with this result. In here, they are showing us interleukin-1 beta and interleukin-6 levels. So here is in serum, and here is just in liver, and that it's the mRNA that codes for the respective interleukin. So for all of them, we can prove that the group that had the Wuzhi capsule would have reduced levels of interleukin, which we can conclude that interleukin levels clearly decreased with the presence of the Wuzhi capsule. This is the Western blot result. So here they are showing us different proteins. These two are controls. And these first four are going to be more present in the group that had the Wuzhi capsule. It's a little bit hard to determine, but if we center attention here, we can see that it's a little bit more thick. So we know that PPR is needed to reduce triglycerides levels, and also that NCAD and LCAD are enzymes needed for the beta oxidation. Therefore, it makes sense that they will be more present in the group that had the Wuzhi capsule. But for example, P65, which is needed for pro-inflammatory cytokines effects, is going to be more present in the group that didn't have the Wuzhi capsule, which also makes sense. And here is just a graphic that is helping us to prove this point, that PPR alpha is going to be more present in the group that has the Wuzhi capsule. Discussions. Hassan said, PPR gamma levels have been found to be increased in the livers of NAFLD mice, and this was positive. Wang said, PPR gamma also has an anti-inflammatory role, and it inhibits the production of various inflammatory cytokines, and this is also positive. And Ouyang said, in the current study, we evaluated the levels of interleukin-1 beta, interleukin-6, and tumoral necrosis factor alpha to assess the MCDD-induced inflammatory state and found that the administration of Wuzhi attenuates the expression of these pro-inflammatory cytokines, and this was also true. For conclusions, we can see that Wuzhi capsule has an effective anti-inflammatory effect because it can reduce interleukin levels and can increase PPR ones. And by this, we can also conclude that the Wuzhi capsule will be an effective treatment for NAFLD patients due to its anti-inflammatory effects and organ recovery properties.